Hi everybody, I already said hi, but I'm Anna Gårdmark and I'm at the Swedish University of Agricultural Sciences. We're all over Sweden, but the center is placed at Ultuna, which is in Uppsala, and I'm at the Coastal Research Laboratory on the coast outside of Uppsala, in a town called Öregrund. So what I will talk about today is why size may matter when you look at ecological communities. Size as in body size. And not only why it might matter, also if you care about it, how you may model it, and what consequences we have of that model choice, what those type of models are good for and what they are not good for, which is sort of preempted this slide. So yeah, the effect of size-dependent processes, not only between individuals or between species, but also within an individual. Why is body size important? And why is it important for the dynamics of populations, but also whole communities, how species affect each other. And then I want us to discuss a little bit, because it's afternoon and I think not to fall asleep, you don't want to listen to me all the time, so keep your eyes out for any benefits or shortcomings that you see of this type of modeling when we go through it. And please, I'd love if you interrupt me with questions, because that also means that you're awake, and that becomes a little bit more interactive. Okay, so my idea, now that I didn't know you, so but when I saw this, well, heard about this course from Maria and Susa, my idea was that after a course like this, well, after my talk, you'll hopefully know something about how body size might influence populations and community dynamics, and you've heard of this approach that I will tell you about if you're awake, and are able to discuss pros and cons of it. But also after the course as a whole, you have got this palette of very short introductions to a variety of models, of modeling approaches. So hopefully you have some idea of where to start looking if you get interested in doing modeling, or if you want to change your approach that you're already using or complement it. If you have a completely different view of what this course is about or this talk about, you can shout and tell me. Then we might know this. Okay, so why does body size matter? Well, if you think of an animal throughout its life, almost all things, I don't know much about microbes, but most animals, they increase in size during their life. And there's no ontogeny, right? Different things happen throughout the life. You don't eat the same thing when you're small. Here's supposedly a herring that I've drawn, which eats zooplankton when it's small, some type of copepods there, and some nectobenthic things when they get bigger, and they might even eat fish, actually. And also, you're often not sexually mature when you're born, but you also have to grow a bit before you can start reproducing. So body size matters for what you do. It matters for what you eat, how you reproduce. And also, if we take the view of this prey here, well, your body size also matters for the risk of being predated upon. And by who? So if we keep on having the perspective of a herring, you don't risk being eaten by a cod if the cod is a larvae, but of course by a larger cod. Okay, so it matters not only for what you do in your own life, but also how you interact with other species. Now, so for example, in fish, for example, mortality, the risk of mortality is often highest when you're small, because the predator is often larger than the prey, and risk of mortality decreases with body size. But it of course depends on the prey of your focus. If you're thinking of what different prey eat, you might also have a different size selective pattern and risk of mortality. If we take another Baltic Sea example, grey seals, whose favorite food in the Baltic is actually herring. Now, here's the Bosnian sea herring, the size distribution, as we've measured it in acoustic and trawl survey, so a research survey. So that's not really the true population, but it's our best sample of the population size distribution. That's this shaded area here. And the black dashed line is what the fishery is taking, the trawl fisheries. So sort of very similar size distribution as to the population. And these are, the red line, are the sizes of the herrings that we find in the grey seal stomachs. So they clearly have a preference for larger individuals than are found on average in the population. So here you have a different perspective, that you might be, from the perspective of a herring, you might be, when you're smaller, you risk being eaten by a cod. While you're growing in size, you might risk being eaten by a larger predator, like the grey seal. Okay, but that's between individuals. Now, body size might also matter within an individual. So this is still between individuals. We have the body size, the mass of perch, which are the black dots, and roach, which are the white dots, and their attack rate on Daphnia of a particular size, which I've now forgotten. So these are experiments. And you see how the attack rate first increases with size, because they're able to handle this prey, but then it declines with size again, because the prey becomes too small to be visible or tractable to handle. Now, so size matters for how they eat, but also matters for the energy that is needed to maintain themselves. So the cost of metabolism, the energetic cost of metabolism, also increases with size. Usually something like this. Okay, so body size matters for what you eat, how you risk being eaten, how you reproduce, but also how you use that energy within your body. Both for how the costs of maintenance here, but also for how you allocate your energy. If you're an immature individual or a larva, you won't allocate your energy to producing gonads or reproducing, for example. So this fact that size matters for what you eat and how you allocate and use your energy means you have a feedback, right? So body size affects my net energy gain, which in turn you allocate to growing, which again affects your body size, right? It's sort of an obvious feedback, but it's an important feedback because it means that the performance of an individual is also influenced by its body growth. And performance in terms of how I grow and how I reproduce, for example. Now this net energy gain is of course eating of someone else. So this is really species interactions feedbacks, right? Here's one species or one individual, a feedback of body size, which in turn affect another species, which is the prey. Okay, so if you start accounting for how things depend on size, there's a number of feedbacks in your system that might affect the outcome or the dynamics. So the framework that I want to present is one that's particularly developed to account for how body size influences population processes. But they don't do it by modeling population processes. They do it by accounting for how the performance of an individual depends on size. And I'll go through that. It's called physiologically structured population models, which is sort of doesn't come easily in the mouth. So it's commonly called PSPM. And they were developed by Dutch guys, Hans Metz and Udo Diekman. And it's been extensively further developed and used by André de Roos and from the Vrije University of Amsterdam, and Lennart Persson in Umeå. And Tom mentioned them yesterday already. So much of what you will hear is based on their work or work that we've done together. So these type of models, what they do then is, as I said, they account for how body size affects what an individual does, right? And that's what you assume. So the resource use, the metabolism, and how you allocate the energy that you've eaten is all size dependent. So you model the state of this individual and its performance, and you track the environment that the individual is in, and then you sum up all the individuals to a population. So you don't model the population per se, but you sum it up. But the population is really the environment for whoever is in that population. So that creates some feedback. I will try to illustrate that in the next slide. I just have to keep track of coffee time. Okay, so what we do, here is the individual level. And what you assume is in the model is that the energy for the individual increases with food intake, so eating, and you have some conversion of how that eaten energy is converted into your own body mass or energy that you can use, while you take away the energy that's needed for metabolism. And what's left is some net energy that can be used for the individual. And that's allocated to, once you've taken away metabolism, it's allocated to growth and reproduction. And all these processes depend on body size. Okay, but they don't only depend on body size. They, of course, depend on what's in the environment, because you're eating things from the environment, right? So food is something that comes from the environment, a resource or a prey or something. So you also assume some dynamics of your resource that's in the environment. So that's affecting the individual. Now what you then do is that you sum up all your individuals, or types of individuals, to a population. So you don't assume any particular dynamics for the population level. And that's a key thing in this approach. What you assume is always on the individual level. And then you sum up and get a population. But all individuals together, of course, have an impact on this resource, right? Because it's being eaten. So you sum up the eating by everybody and how many they are, and that, of course, affects the resource, which in turn is going to affect how much food this guy gets in the next time step. So you have this feedback between what an individual eats, how it allocates it and grows, which affects its own size, which will affect what it eats, and how it survives and reproduces. So you sum all that up, which in turn affects the resource, which in turn will affect itself. So this feedback that this creates, it's sort of intuitive if you're in dynamic models, that feedbacks that can create all kinds of interesting dynamics at the population level, but also at a community level, because resources are really just other species in your system. Yeah?
Can I picture that as a logical terror model, but in there you look at the population itself without taking into account that those species, they grow, and then they might also eat, maybe each individual eats them also more, like there is a certain dynamic. But
yeah.
What is this individual level box doing in addition to this kind of?
Well, in a logical terror model, first, you don't have any size. So you assume that they, if anything, they're all having the same size. So there's no variation within a population. So that's what you're getting here, because you have individuals of all kinds of sizes that you sum up to a population. And the other important thing is that you're assuming, at the logical terror, things about how a population works as an entity. You're not saying that processes happen at the individual level, but it's never a population that eats another population. It's always an individual that eats another individual. So that's sort of the conceptual differences. So what this means, that the fact that we assume things here and assume things here, but we don't assume things at the population level, means that whatever comes up here is emerging from the model. That is a result from the model. And what that means is that we can test these against data. It's not like we first fitted a population model to population data and then wanted to test it against population data, right? Because this type of models are parameterized from experiments done on an individual level. For example, like the perch roach attack rates you saw before. I'm pointing up here because it used to be here. Right? So you assume things at the individual level. That's also what you parameterize it on laboratory experiments often. The only place currently where we assume things on the population level is if we don't do a detailed model for the resources. But we can, of course, instead of having aggregated dynamics here, also do individual level models for the resources. And I'll come back with examples of that. So the key thing is, what comes out at the population and community dynamics level is emerging from the model. Okay.
So basically it's a bit of a similar approach, even though being very different from measurements, but you can study the results the similar way with the interactions. Or
Yeah, dynamic modelers are very keen about emerging results from the model.
okay.
Meaning things that, yeah, so in that sense, yes, because he had emergent patterns of functional responses, for example, which means that he can then compare them to data on functional responses.
Yeah.
Yeah. Yeah. So yes, emerging properties are good because we can compare them to data, which is a nice walk over to this slide, which is an example from a model, one of the more detailed models of a single species perch model by David Claassen, a former PhD student of André de Roos, and some data. And this is actually also exemplified in, I don't know if you had time to do any of the readings before, but in this de Roos-Persson paper that I sent out as a reading, this is also exemplified. So what comes out here is also then that the growth patterns of the individuals is also an emerging result from your model. Remember that feedback? So we weren't assuming any particular growth shapes because that resulted from what they ate and how they allocated their energy and what was left, right? So what he found, what David found in his study was that you could have this interesting pattern of alternating states with quite stunted individuals. So this is how an individual grows, or cohorts actually. This is time, and this is here is a cohort is born, a year class is born, and this is how they grow in length. I don't have centimeters here, sorry. But every now and then, when a cohort is not very severely eaten by older cannibals, they can escape cannibalism and have a very rapid growth. And that means that when they have a very rapid growth, they're much bigger than whoever is born afterwards. So they can use them as to prey upon because they're cannibals. That means they get a lot of energy from these conspecifics, and then they can use all this energy to grow to giants. So he found this pattern of sometimes you had dwarfs and sometimes you had this giant perch. And that's actually something that you see in lakes in Sweden. In Swedish it's called tusenbrödrasjöar, 1,000 brothers lakes, where you have these lakes with really stunted, lots and lots but stunted perch. And here's one of the favorite lakes of Lennart Persson up in Umeå. He has 4 lakes that he used experimentally since the 80s, and they're all called abborrtjärn, the perch pond, basically. Number 3, there's 4 of them. And this, in number 3, there is only perch, no other fish species. So the only fish that they eat is perch. They're cannibalistic. They eat other things than fish, then, obviously, but they eat zooplankton and bedbug too. And what he found was actually the same pattern. So this is observation data on size trajectories of cohorts in different years. And you can see that in 88, some of them took off and escaped cannibalism and could become these giant patterns. So that's why modelers like patterns that emerge from your model. If I had now used, think of the reverse. If I had built the model, say I'm interested in giants, let's take some data on giants and build a model. If now I would have assumed this pattern in my model, of course I can't test my model result on this data because I already assumed it, so it's not a proper test. So things that emerge from a model is nicely testable against data. Okay, so that's the sort of the general idea of the model, is that you assume things at the individual level, and you assume how that depends on size and on the environment that the individual is in. And then you sum up all the individuals to a population, which in turn affects the environment. So you get these feedbacks and emerging patterns.
I have a question regarding the second plot there.
Yeah.
We see that the line starting from 84, 86, 88, no, not 88, but between 84 and 88, they stopped at some point.
Yeah.
So that means that the fish were dead?
Yeah, just like here, they also dead here.
Okay, but so they followed individual fishes, so they were tagged somehow, or am I missing something?
They followed, well, no, they're not tagged, they're actually killed. So this is, so the standard deviation here is the variation between individuals in their size at age. So you fished up individuals and you determined their age by their otoliths, so the bone structure in their ear. So this is the average for a cohort. It's not an individual. One individual.
And so that's the leg where there are only perch.
Yeah.
And did they remove the other fishes, or
no? It's naturally. It's
natural.
Yeah. And some of the other legs, they did a lot of interesting introduction experiments.
The actual data doesn't really fit to the model, because we see that after 88, well, the following cohorts don't display this lag in the growth.
That's true. So this part is not represented well.
Which
now you can
is critical for the assumption that these guys draw as to giant because they are eating their compartment.
But not
well, it's not really this part that's critical. It's actually this part that they can take off rapidly because this part that they escape these ones in predation. But I mean, of course, you can always find. It depends on how if you want to, for example, if you want to make quantitative comparisons, you can also find things that don't fit super well.
No.
So it's a question of, but that's an important question in general, in modeling in general. What is it that you want to compare with data from your model? Is it a general pattern? Is it the fact that you just found a regime shift, for example, since we've been talking about regime shifts? Or is it the fact that you find giants or dwarfs? Or is it the actual levels? Or like here, is it the mechanism? Do we want to make sure that we catch the same mechanism? So just like you have to, like Carsten said, be aware of what you're modeling, you also have to be aware of what is it that I want to compare. So that's a good point. Okay, so now since you started talking, we can continue talking a little bit. So can you see any benefits of this approach and any drawbacks of this approach? And I can make a list and we can see. You can probably come up with many more than I can. Yeah?
I guess, like you said you had to estimate or assume the food intake and what was it, ingestion rates?
Yeah.
That would maybe be uncertain if you don't have a very good. Of course, you can estimate in the field, but it's not applicable to.
Yeah.
And also, if this is a very variable parameter, like over the course of the year or something.
Yeah. So they're both uncertain, and it's a question about what you measure in the lab is applicable to a field situation. Yeah, that's a very good point.
Is that was another? Is that the intake or the consumption? Was that just a factor between 0 and 1? You multiplied with some. What was it? The energy conversion.
Energy conversion is in these models a constant factor, but food intake is a function of both size-dependent attack rates and size-dependent handling times or digestion times and things like that. Yeah. So and yeah.
Since you're looking at individual.
That was a plus. Sorry. Yeah. It's supposed to be minus.
Since we are looking at individual within the population, we need to sample much more because.
To compare output, you mean, or just to parameterize?
Just
yeah. Parameterization.
Do you guys need or should we put a slide
Oh yeah, thank you. Yeah? Anything else? Yeah?
I believe there are population dynamics which should be included.
Sorry, say
I
again?
think there are population dynamics which are not taken into account.
What type of things?
Like density dependent, like
good
when
question.
individuals are competing, the more you have, that has also made an effect.
Yeah, that's actually an emerging thing. The density dependence emerges, right? Because of this very feedback. So one individual does not impact the resource, but the sum of all the individuals, which is the density, affects the resource and limits the resource for this particular individual. So the density dependence actually emerges in the model because of this loop, feedback loop.
You just said it's a factor of size rather than of numbers.
Well, you do, numbers is here because you sum up all your individuals.
But this is
You're just
probably
a
a
comfort
linear effect there. You say like the higher the population is, or not? Is there?
No, I'm not assuming a linear. Well, yeah, it depends
Well,
on what you
that's
assume
more
how the mortality
more-
looks here, of course. Yes, it will depend on how you assume that the mortality here depends on. No, it doesn't. Yeah, sorry, I was going to say something that was not correct. I mean, you sum up what all the individuals eat and remove that from here. And that means that this becomes limiting, unless you have assumed some really strange resource dynamics. So, I mean, if we take classical density dependence, now I make a deviation here, right? You're basically your birth rate per
But I think
individual.
like the individual behaves differently and takes up differently if there are more competitors.
All right,
This is
yes,
like what
okay.
kind of
So what's
dynamic
not
of-
here, yeah, okay. So there's one thing that's not here, and that's interference competition, for example. That part is not here. Exploitation competition, which means that, you know, you get less food in a population that is highly, where there are a lot of conspecifics because they eat the same thing. That is in here via the feedback. But the interference competition is not. Like if you and I start fighting over the soup for lunch today. That part is not there. Yeah. Maybe over the chicken or the apple pie. Okay, so yeah, okay. There might be, anybody want to come up with something positive? I probably listed them already because I'm biased. Population processes. And still not a, yeah, it's still something you do between individuals. So if you included it, you would include it at the individual level, I think, in this framework. But interference competition. Yeah?
This is probably a really stupid question.
Yeah.
How do you get the right population levels from the individuals? Is it determined by the resource at each time, or
The right, what do you mean the right?
like the population? Because then you need to have the right population levels in order to have the right effect on the resources, basically.
But what's right? I don't
No,
know
but
the
like
right part.
because eventually, when you compare it with data and things, like the advanced levels or the
That's one of the drawbacks often with this approach is that the quantitative levels aren't always comparable because they very much often depend on the what you assume here in terms of productivity,
okay.
for example. So that's a general drawback in terms of it's easier, for example, for a statistical model that you already fitted to your data that you've been out sampling, which will give you a model exactly at the right scale.
Mm.
It might not capture the right processes, but it will be captured the right scale. Yeah.
But if you have the right or the real resource availability plus realistic food intake and then you've got the conversion rates and such, you could assume that you would get similar levels, population levels than in reality. Or
Yeah, but you know what food webs are like. You know, the resource. There's usually more than two things in the resource, and that's actually if we jump to. That's actually one of the drawbacks here. It's not only that it's hard to parameterize because you have a lot of parameters, but the fact that you have a lot of parameters also means that it's hard to analyze when you're trying to gain understanding, when you try to look at how does this particular parameter influence my results. A sensitivity analysis, for example, or some type of analysis. So that becomes hard when you have a lot of parameters. But it also means that both because it's computational demanding, but also because it's hard to understand when you have too many parameters at once. There's a limit to the number of species that you model with this approach. So we've had 3 species where you account for size and the number of resources that you ignore size in, but not more than that. So we're very far from, you know, like an ecosystem model of the Baltic where you have 22 components, or a trophic network where you might have 100 species in a food web, for example. Or we're not even at the 7 species of the Barents Sea. But there are other things, like, for example, you had these emerging things, that makes it very nicely to be testable against data. And another thing is actually that because we're looking at things at the individual level, we can and have these population patterns emerge. We not only can test this emerging population levels, but we can also track backwards. If I have this particular population dynamics, what are the underlying mechanisms at the individual level that in my model causes this pattern? So it's something that you can use to try to understand mechanisms underlying particular population dynamical patterns. I'm actually I promised I would give you some evolutionary model talk. I have such a talk, but I think to not exhaust you, I think we rather split this talk in pieces and take a coffee break soonish. I have, what do we have, 10 minutes. So let's take one little slot more.
Also, if you wish, we can go earlier and come back earlier. It's up to you.
We take a 10-15 minutes more and then we take a coffee break. Because you've been talking, so you can listen a little bit now. Okay, so let's now look at how, so the previous example with cannibalism perch, we saw that assuming that processes at individual depend on size actually affected the population dynamics that came out, or the population structure that came out at least, because you had these dwarfs and you had these giants, right? So now let's move to more than one species and look at how size-dependent processes can affect the patterns that you see in communities. And communities here then are 2-3 species, so not huge communities. There's a pike here waiting to feed on whatever swims by. Okay, so here's one of the classical examples from this particular framework, which Tom also mentioned yesterday. It's a physiologically structured model that includes predator and prey. And here it's actually the prey where you look at the size structure. And you have a predator in which you ignore the size structure. The only thing that they assume, so this is work by André de Roos and Lennart de Persson in Proceedings of National Academy of Sciences. The only thing that they assume is that for the predator doesn't feed on all roach, the perch here. They feed on small roach and of a particular interval, right? So they don't feed on the very smallest ones and not on the larger ones. So with this assumption and the standard assumption of that what happens to a roach depends on its body size and on what it eats, right? So its growth, the growth of the roach depends on its resources. They actually find this case of alternative stable states. You remember alternative stable states, like in an identical environment, you can have two types of communities here. And you can't say which one you will get, depending on. It will depend on the initial conditions. And what they find is states with and without the predator in the system. And this is what they call the emergent allele effect. And I made a little comic illustration to try to illustrate that. So in a system where there's no perch, the life cycle of and the population of the roach looks something like this. So you have mature individuals, not very many, that produce some few youngs. And they are still few. Also this vulnerable stage aren't very many. But there are many enough to compete for resources such that these guys, they don't grow very much. They're all not stunted, but very few of them gain enough resources to mature and start reproducing. So competition is high here, and they're all resource demanded. So what happens if you have perch in the system and enough of the perch such that they can actually initially press down the number of the vulnerable stage, is that, well, since there are fewer of them than there used to be, these guys, they don't compete as much for the resources any longer because there are fewer of them, right? So they're released a bit from the competition, which means each of them gets more food. If each of them gets more food, more of them can reach the maturation threshold, become big and mature or reproduce, and get more offspring. So the thing in this model is that this increase in their maturation rate and the output of these big guys more than compensates for the mortality on these guys. So the fact is that by grazing, by feeding on these guys, they're actually producing more of their own prey of the vulnerable stage. So the whole phenomenon that emerges requires overcompensation in the prey dynamics, right? So they need to compensate, more than compensate. Thereof the term overcompensate, more than compensate for the mortality. Okay, so that means that if you lose perch from a system, it goes back to here, and they actually can't invade because these are too few. Their vulnerable stage, the prey are too few. And that's why you could end up with this alternative stable states, with and without the predator in the system. And that was called the emergent allele effect. That's what they called it. So that's the thing where you can see how the size-specific predation on a particular size and the feedback on resource-dependent growth of these guys and size-dependent growth creates these two different types of communities. So it does matter. You don't see this if you don't account for the size-dependent processes. And I think I will give you another example after coffee. So I think we stop here because this is a longer example with well, I should say, sorry, I go back. This, since we're in the promised Baltic examples, there is perch and roach in the Baltic, by the way, in the coast, but it's also been shown for a related model by one of Andre's students that was parameterized for Baltic cod and Baltic sprat. So she also found this emergent allele effect in the Baltic cod, Baltic sprat. And for an exploited species, it of course matters a lot. If you fish away the cod or the perch, you might not get it back if you have these type of feedbacks. Okay? You might not get it back for other reasons, like the changing environment that Torsten was talking about, but here's an internal community dynamic response that made it not go back. Okay, so here's another example that I want to give you of physiologically supported population models. And this is built for herring, Baltic Sea herring and sprat by Magnus Huss and me, who did a postdoc with me, and together with Andre de Roos and some other colleagues. And what I want to show you with this example are two things. This is how size-dependent competition affects the composition of this community, if you wish, and how it responds to environmental change, and how size-dependent competition can lead to alternative stable states, also in this type of systems with two competitors. And the third thing is really that how we can use this type of modeling approach to understand the mechanisms that causes the alternative stable states. So when we model it, it's not just like turning the crank and cool result, alternative stable state come up. But it's like, how do you use this model to try to understand the mechanism that causes this community dynamic or response? So in this case, we have fully size-structured herring and a sprat, which is what it's supposed to look like. And again, then what they do throughout life depends on their size. The things to notice is that sprat in this model matures at 9 centimeters. When they reach that, they start allocating energy to the gonads. And when they have large enough gonads and they are in the right season, they will reproduce. Whereas herring, they mature at 15 centimeters. So they're larger. And they compete for shared zooplankton resources. So we model two different zooplankton resources, and we vary how much they share these resources, how much they overlap in their resource use. And in the model, herring also have this nectobenthic, these mysids, as their own exclusive resource. But I won't talk much about the benthic part of it. So the focus is really that we vary here the overlap that we have in resource use between them. Okay, so to the first thing that I want to make you remember then. So because we're assuming that attack rate and handling time and metabolism, this cost, so energy cost for maintenance, depend on size, the consequence is that the competition depends on body size or competitiveness. So what we have here on the y-axis is the critical resource level. That's a density of resources that an individual of this particular size needs to sustain itself. That's the minimum that it needs. Now, so what you see here is that if you concentrate on herring, for example, the blue line, this critical resource level increases with body size. So a small individual can always sustain on less resources than a large individual. So competition within a species depends on, or within a population depends on body size, but also between species. So if we use the best data that we have on spread and herring, which aren't super good, that is to be admitted, we see that herring is actually for the same size, herring can sustain on lower resources than the spread. Okay, so for a given resources, the herring actually grows faster than spread. Now, at a population level, spreads usually don't grow very large. They are over here, whereas herring grow larger. So on a population level, the larger herring individuals, of course, do worse than a small spread individuals. But for the same size, herring can sustain on lower resource levels. Okay, so resource competition depends on body size. Now, this affects the responses of this community. So that's an example first. So we really can have, in this system, we basically have two sort of competitive strategies. Either we have this herring, which is really can sustain on lower resources than spread for the same body size. But spread has another advantage which herring doesn't have. It matures earlier. It only has to grow to 9 centimeters of size, and then they can start reproducing. Whereas the herring had to be 15 centimeters. So how does this, can we find a balance in this system? Well, in the Baltic Sea we have both spread and herring, so I guess you will figure out that the answer is yes. The question is how that changes with a changing environment. So this is a sort of a complicated picture, so I ask if you don't get my explanation and I will take it slowly. So what we have here was plotted is the resulting community that you can have for a given environment. So this is the productivity of your zooplankton resource. So here's a very rich system with a lot of zooplankton or high productivity, and here is a poor system. And here is the degree to which herring and spread share resources. So over here we have single species system. They don't share resources at all. And up here they eat identical amounts of the two zooplankton resources. So we vary competition, you can say, or resource overlap here. So what we see is that if you have a really poor system, too low zooplankton productivity, you don't have spread or herring because there's not enough zooplankton in the system to sustain them. Now, if productivity increases, you get a system with only spread. And why is that? Because we said herring could sustain on lower zooplankton levels than spread could for a given size. The thing is that with these levels, there's not enough energy for herring to grow to 15 centimeters. So they can't reproduce, so you don't get a herring population. But there is enough resources for spread to grow to 9 centimeters. Okay, so at low resource level you only have spread. So this is the winning strategy at this low resource level, the early maturation. Now, for a larger region, if you have a single species system or not much competition, so all in this region, you get a system with herring and spread in it, so a coexistence. But if you have really high competition, say fully resource overlap or somewhere up here, if you increase productivity, the herring will actually outcompete spread. And the reason is that herring at these levels of productivity, herring grows really fast to the 15 centimeters and produce a lot of young, which are small, and outcompetes the spread that are of the same size. Okay, so if we have size-dependent competition, high competition level up here, the responses of a system to enrichment will be different if we're up here with a lot of competition than if down here, right? Here, enrichment leads to loss of spread, whereas here it doesn't. Okay, so it determines how the system responds to productivity. I'm not showing it. I can show it later if someone's interested. But it also determines how the system responds to mortality, or you can say differently. For different levels of mortality, you get different responses to the environment as well. And that also depends on the size-dependent processes. But that's a little sidetrack. Okay, so the other example where it matters is this example of how size-dependent competition can result in alternative stable states. And alternative stable states, you've heard it so many times now. Okay, the two types of states can occur in an identical environment. And if you push the system beyond the boundary that separates these two states, you might end up in the other state. So what we find in the spread herring model for certain levels of mortality is that when we increase the zooplankton productivity, we can actually have two different states. State 1 and state 2. And red is spread and blue is herring. And this is density there on the y-axis. And I will walk through this as well. So first you have this system where we said before, low productivity, only spread, and nothing else. So there's no alternative states there. But in this region, we have alternative states because we can have either a spread only system, which is the red line on the top, state 1, or we can have a herring only system. And the reason is that whoever is in the system first also determines the dynamics of the resource. So the dynamics of, in this case, the spread population induces dynamics in the resource, which prevents the other species from invading. So that's called priority effects. Yeah, we'll leave it at that. So maybe more interesting is this part, where we also have another type of alternative stable states. So you see here you can have a state with a coexistence with both of the species in it, or we can have a system with only herring. And actually, if we end up in a system with only herring, if we make the spread go extinct from this system, it won't be able to re-invade the system. So those type of alternative stable states then have consequences, of course, if you think about exploitation. If you overexploit a population in a system where you have alternative stable states, it might not be able to recover. And I'll go through the reason why we see these alternative stable states in this system. And it all comes to do with this competitive asymmetry. That herring is more efficient than spread for when the same size. You remember the slide I showed you, at the same size. And also the fact that within a population, small individuals are more resource efficient than large ones. And what that results in, that small individuals are more resource efficient than large ones, is that you get cycles in your population. So I'll walk you through this as well. Well, you have herring and spread densities here. Spread is again red and herring is blue, and here these are population trajectories over time. So density is in number of individuals. So when you have a spawning taking place, you have a lot of small individuals, a lot of larvae, that eventually die off. The thing is that when these small ones come, they actually are so efficient that they outcompete their older conspecifics, right? So the total will go something like this. So you get cohort cycles. What you also see is that these cycles, they differ in length. You see the spread has more frequent shifts than herring. And the reason is that the cycle length differ is because it's determined by the maturation age, right? Spread reproduces already at 9 centimeters and herring at 15. Spread reaches 9 centimeters faster, so they will reproduce more often, so you have more frequent cycles, okay? But since it differs with maturation age, cycle lengths will differ between species. So species in a system will have different periodicity. Another thing that affects the cycle length is mortality of the system. And what that means is that if you have different environments with different mortality in them, your cycle length will also be different. Or if you impose mortality, say you start fishing, or there's an invasive predator in the system, your cyclicity will change. Okay, population cycle. So why does this matter? Well, it matters because it gives cyclic resource dynamics. So there's no external force or climate force or anything that makes this cycle happen. It's just predator-prey cycles between your fish and your zooplankton. So the green here, I just added the lines. You have the blue herring and the red sprat as you had before over time, and now you have the zooplankton dynamics. And what you see is that if we have only the single species system, so only herring, you get a cycle in the zooplankton that has the same periodicity as the herring does. And the same goes for when you have sprat. The periodicity in the zooplankton follows what's in the fish population. And this is basically because the resources are just driven by the predation pressure from the fish species here with a constant inflow. So what that means is that if we have a single species system with only herring, and we have sprat trying to invade this system, it's not going to invade an environment to what it's used to, or what it would have if it was the only species around, which is this environment. No, it's trying to invade this environment, right? Because this is a herring zooplankton system in which we're trying to recover the sprat or invade the sprat, which is quite a different cyclicity or periodicity than the sprat itself. And what happens then is that it's actually too out of phase of the resource. So here's a sprat trying to invade a herring population. And since it's out of phase with the resource, which is set by the dominant competitor herring, it goes extinct. So it can't cope. Whereas if we have a sprat system only, herring can actually cope with this environment, although it's not its own. And that's because it is the stronger competitor or the superior competitor. So despite having out of phase and low levels, it can still do better than the sprat. Okay. And what you also can see is that, so there's a lot of graphs, and soon you're tired, I think. Here's a system where, I don't know if you can see it, it's both blue and red. So here's the coexistence system with both herring and sprat in it. And when you have the two species in the system, the zooplankton dynamics changed. So you had a system with only sprat, you had a herring invading, and this is the dynamics you had in the zooplankton then. But when you have both species around, your dynamics of the zooplankton have changed. So but it's still not the same as you have in a single species herring system. Yeah, question?
So,
yeah, you first.
Okay, that's fine. It's just in herring suddenly, like the period is much smaller now. Or?
Here it's smaller than here.
No, the blue under the red.
The frequency of the population.
Yeah.
Yes.
Suddenly.
Yes, it shifted. Yes, that's true. So it's adjusting too, because the resource level is also high for them to reproduce faster. I don't know if that's possible to see. Is it?
Or is that an artifact?
The average? No, it's not an artifact. So what? I don't know if that's easy to see. I should have plotted the average resource level as well. What you see here is that it, since here, okay, we go into details. So sprat is not as an efficient to press down the resource as herring is, right? You remember that. So that means that the resource is recovering quite fast after this peak of the small sprat that was feeding down on it. You see, it's really picking up fast. Whereas here, the peak is fast, but it doesn't come up as high, and it's a slow recovery rate. So the average zooplankton level is actually higher here than here. So there's basically more zooplankton around, so herring can reproduce faster because they grow fast at the 15 centimeter when they mature.
Okay,
so they get a faster,
zooplankton.
yeah.
Okay.
But that's very observant. That means you were actually looking at the graphs. So there one more question. Do you have a question? The same question. Okay.
I have a question adding to it, but then shouldn't the plankton respond differently now if also herrings invading the system and puts additional pressure on?
Yes, it does. And that's exactly this part. It's just that when they're invading, there are so few.
Okay.
So when you see them, when they start having an effect, it's over here. Yeah. But that's a good point. And that's actually another thing that you can see is that you sometimes see that they can initially invade, but once they themselves are so many that they start having an effect on the resources, they might also go extinct if they have the wrong effect on the resources, if they overexploit the resources, basically. Okay, so the whole idea is that if you have these population cycles, which are created by this size-dependent competition within the population, just by the fact that small guys are more efficient in using the resource than large guys. So small individuals outcompete large individuals. You get these cycles. And cycles in the fish populations give cycles in the resource. And the resource is the environment for all the other fish around. And if that environment is too hard for them to invade in, they won't be able to. So that's the mechanism here that gives the alternative stable states in this particular system. And what it also, so this particular mechanism is something that has not been shown before. So it basically says that if you're a poor competitor and you're cycling, but you're cycling out of phase with a stronger competitor, you will be unlikely to recover because you will be out of phase with your resource. Okay, so that, yeah, go ahead.
I agree with, well, okay, I just have a question regarding the scale of the cycling of zooplankton, because it's like, it's quite wide.
Yes, it is. And it all depends on what I assume for productivity level. Well, not all, but for the productivity turnover rate of zooplankton,
it seems
and also the attack rate.
I'm not a professional zooplankton or plankton person, but
Me neither.
it doesn't follow annual pattern.
No, there's no seasonality, so it
Okay.
shouldn't follow annual patterns.
Okay.
There have been, so one of Anna Gårdmark's PhD students, she did seasonal models. Actually, Magnus Huss during his PhD also did seasonal models, so where you can add those complexities if you want, but you're absolutely right. Right, here it's nothing. Here it's just, you know, between years dynamics.
So it's just pure water.
Yeah,
Okay.
it's basically back to this, right? I wouldn't go out comparing that zooplankton data with quantitative real data on zooplankton. I'm after showing a mechanism that creates alternative stable states in this particular system. I will show you some data now, though, but it won't be zooplankton. So, I mean, this has all been models, and it's, well, we know from data that individual processes depend on size. You saw the attack rate functions over size in the lab and things like that. But can we see these patterns of cyclicity that we think can lead to alternative stable states? So let's have some Baltic examples. What you have here on the top is cod predation mortality on 1-year-old herring and sprat. And again, herring is blue and sprat is red. So when and you have a time axis here. So when the cod basically disappeared from the Baltic, of course, predation mortality went up. So we have these two different environments with high mortality and low mortality, right? Remember, cyclicity was affected by mortality in our model. So the question is, does it affect the cyclicity of the Baltic Sea herring and sprat? Here's the abundance of sprat and herring 1-year-olds for the same time period. Now, if you fit autocorrelation functions to these time series for the different periods, you don't find any significant patterns because the time series are too short. I did that. I really wanted to find a pattern, believe me, but I didn't. But if you dare to fit by eye, you do see some more frequent shifts, at least in the sprat, than you do here. But that's not an evidence of anything. It's just a fit by eye pattern. What we do see is, if you look at the bottom graph here, is the average age at maturation. Or not, sorry, not the average. The first age at which they reach the sizes that we in this model said that they were maturing at. Right? So the red is sprat. It's still maturing at, well, they're still reaching the sizes already in the first year of life. Whereas for herring, it seemed to have shifted. We more frequently have a higher age at maturation now than we had in the cod period. We do have some outlying years, so it's not a super clear pattern, but we see a shift, could see a shift in age at maturation. So we basically have these two periods. You have this high mortality, where you have somehow irregular dynamics, and where you also have this, here's another maybe tricky graph. So what I did was that I correlated juveniles and adults. So I wanted to see, are they are they high, the biomasses of juvenile adults, at the same time? Because if they are, I don't have these cohort cycles, right? When you had a lot of young, they were outcompeting a lot of small ones, they were outcompeting the large ones. So if they co-vary, I don't have these cohort cycles. And that's really what we see here in the high mortality period. So the dark dots here is the high mortality period, and the open dots are the low mortality period. So for herring, the highest correlation is really when I have no lag in time. They are positively correlated at the same time, right? And the same goes for spread. The highest correlation I find between juveniles and adults are when they are in phase, so they're not out of phase. Okay, and I don't really see a difference in herring and spread age at maturation, which could mean that they were, if anything, they would cycle in phase with each other with the same periodicity. Now in this low mortality period, I have this, well, at least I have these strong cohorts and weak cohorts by the looks of it. And I also can see that when I look at the correlation between juveniles and adults, because now the highest correlation, so the highest open circle here, positive correlation, is when they're out of phase, juveniles and adults. So we see that for herring, when they're actually out of phase with a 3-year lag, and for spread, when they're out of phase with a 1-year lag. Ideally, I would want to find, you know, a lag that fits with the age at maturation. That would be really neat. It doesn't really here, but okay. But anyway, it indicates that we have strong cohorts and followed by weak cohorts, strong cohorts followed by weak cohorts, and that adults and juveniles are out of phase. Okay. So what does this really show? Well, it could be driven by other things, but it can also. But what it does there to say is that we see the cycles. We see cohort cycles. We see these out of phase dynamics and strong cohorts followed by weak cohorts. So they are cycling, and they are strong competitors, spread and herring in the Baltic. And we have this difference in age at maturation, saying that, you know, they might be cycling out of phase. I can't say that we have alternative stable states, right? But I know that these type of mechanisms that I use the model to elucidate, and these mechanisms that in the model causes alternative stable states, I see some evidence for that in the data that those mechanisms might be there. Now, I showed this in a talk not long ago, before Christmas, to people who know a lot about these pelagics and zooplankton and things. And they go, yeah, I like your model, but you read the data like the devil reads the Bible. And that means that, of course, in reality, which I don't have in the model, is of course these are all affected by a lot of climate drivers. They're affected by fishing. They're affected by a lot of things that I'm not looking at right here. So there might well be other things. But I do see the competition, I do see the periodicity, and I do see this change, difference in maturation age. So, yeah, go ahead.
With this rat herring relationship, earlier you showed that herring is more, what do you say?
Can sustain our resources.
Competitor, or kind
yeah.
Of, yeah. And then in the Baltic we have seen the opposite sprat taking over in the...
Yeah, so what this shows is that herring is better if they are of the same size.
Okay, so...
Right? But at the population level, since spread rarely grows to more than 18 centimeters, it will always outcompete herring that are, well, say about 22 centimeters or so, and herring can grow to 35 centimeters. So at a population level, spread might do much better on resources. Another reason in the Baltic is, of course, yeah, okay.
Yeah, yeah.
No, go ahead.
Yeah. But then there was the one, say who you're working with. Who's
Magnus?
His graph where you say that when there's high resources, it still seems that herring would do better if the zooplankton productivity is higher.
Yeah.
But how does that relate on the situation we have in the Central Baltic Sea currently? Can you say if we're somewhere like
well, that comes back to our discussion on, you know, how do you do quantitative comparisons with these models with reality on things that are on this very general level, like productivity of zooplankton. I don't know where we are in the Baltic. I could say we're probably not over here because we do have spread and herring. I can do Benjamin's version, you know, I can I know where I'm not. I think that's actually quite mind-free, so you should exercise that part more. Yeah, so I wish I could say, you know, and that's also what managers always ask, you know, so where are we and what will happen? They want to say, oh yeah, we're there, right? Or something. But that's not really what these models can do. You can use them to look for mechanisms and understand how things at a population level might be caused by individual processes. So now that you've seen the examples, do you have come up with any more things that worries you? Or you'll go, hooray, I know what I can use this for. Yeah?
It's kind of related to what you talked about with the 2 lines.
Yeah.
I just wonder, let's say the relationship is not the same every year between.
Yeah.
So I was wondering if you played around with different, like these resource lines, if you played around with different combinations and see how you did it. It's probably very tricky, but.
No, so what we did was that we played around with one alternative scenario. So we changed the attack rates of sprat to make it more efficient, such that the sprat and herring lines, they were identical until sprat became mature at 9 centimeters, and then the herring was again better. So we played around with that, but I mean that still was, we used that for the whole simulations. We never did any stochasticity or any seasonality or between year variation or climate drivers on this.
No, but even
so that's
because that's an assumption for running, like you use this as a constant relationship, so it'd be cool to see the round or the variation you would get if you flip the lines or something. Yeah, no, I can't see well, I could do that. I could flip the attack rates every year if I wanted, but I did not do that. Yeah, but yeah. Yeah, maybe.
I was also struck by this difference between, first, by this change in the size, which many processes you could easily think of going the other way. So like specific couple rates go the other way, swimming costs go the other way. What is it based on, that size relationship? That's one question. And then the other one is, what's the difference of the given size? What's the mechanism? If you have a similar sized herring and a similar sized sprats, exactly why will you find orders of magnitude differences in the attack rates?
Yeah, and this, so the thing was, okay, so that the lines that we had with the critical resource densities over size, that's not assumed. That's a consequence of how we assume that metabolism and attack rate and handling time depends on size. So so the fact that sprat is less efficient here than herring is based on when we parameterize handling time. So handling time here is they're mainly limited by the time it takes to digest whatever they have there in the stomach. So when we try to parameterize digestion time, we do it by the inverse. We're saying what's the maximum that they can ingest, and which is the inverse of digestion time then. And by to try to get some real numbers on that, we look at what's the maximum growth that has been observed for very small herring individuals when they're not affected by a lot of other like growth history or things like that. And the data that we had on maximum growth on herring showed a faster growth than sprat does. So that's where it comes from. Here.
Yeah, so it's the growth potential with size, really.
Yeah.
And
which
is species difference.
Pardon?
And the species differences?
Yeah, that is. So when you look at the species, when you look at the maximum growth rate for small sizes for herring and compare it to sprat, it's actually higher for herring. So if you can grow faster, you have a higher ingestion rate, which means you have shorter handling time, which means you're going to be able to suppress the resource to lower levels for the same size. But I mean, these are, and that's another shortcoming that, these models are really sensitive, since everything's driven by the size-dependent processes. They're of course very sensitive to what we assume here, or how we parameterize the size-dependent processes. And there's actually very few species for which they've done really consistent and well-performed studies of how, for example, functional responses or ingestion rates changes with size. So often when we parameterize these models for new species, then you have quite scarce data to parameterize them from. So that's what we talked about before. But the benefit that I'm trying to show here is that you can actually gain mechanistic understanding of the patterns that emerge by going backwards and look at individual processes. The drawback of complexity that we had is has partly been resolved by André de Roos, who showed how you can reduce this model where you assume, where you track the all possible sizes that an individual can have and how the processes relate to that, how that can be simplified into a stage-structured biomass model. Now, then someone that's used to modeling starts to wonder, well, what's so new about this? Because modeling stages were done for ages. That's matrix modeling, basically. Well, what's new here is that he actually derived the flows between, or the rates by which they growth and survival rates, and maturation rates and reproduction rates. He derived them from the continuous model, from this model that is the complex model. And what's so good about that? Well, the good thing about that is that if we trust, how the individual processes here lead to emergent population and community dynamics, they are identical. This model gives the identical result because the rates are derived from this model. So that sounds like hooray! We have this simple tool to still capture all the relevant dynamics that we think occur, or processes that occur at the individual level. And we can use it for more species, and it's easy. The only drawback is they're only identical at equilibrium. So if you want to look at time trajectories and transient and response times and things, they are not equal or identical. But it's good complement if you're interested in the direction of things that happens, or equilibrium solutions, for example. So I thought I'd summarize that part, because now we're back from coffee and a bit tired. So this is the end of the size-dependent thing. So basically what I tried to show you today is how body size of an individual determines what it eats, how it eats, and how it uses that energy that it gains, and how it allocates it. And the consequence of that is that the interactions between individuals and the environment depends on body size. And since that environment is other individuals, either of the same species, if it is, you get population dynamic feedbacks like density dependence, but also the environment are individuals of other species. So that gives size-dependent species interactions, right? So the fact that things we do in our body and with our body with relation to the environment depends on body size, have consequences for population and community dynamics. And we've gone through this physiologically structured population models, the idea of it. This is one of the papers in the readings, but I think they did. Yeah, you sent it now, because I was late sending it to Maria on Thursday, and then it was stuck in all the other organizations. So the concept of the modeling philosophy is in this paper that you got, Anoika's paper. And I also touched upon that these models can be simplified to stage-structured biomass models. And I showed you these different examples of how these size-dependent interactions can determine population dynamics and species or community responses. You saw these alternative stable states in the predator-prey system, the emergent allele effect. You saw it in the competition system. You saw these giants and dwarfs, the 1,000 brothers lakes of the perch, the cannibalism, and all these things. Yeah. And why does it matter at all? It's all in the model. Well, we know that body size determines what we do as individuals. And if it affects the community dynamics, it also affects how these communities respond to all the environmental drivers or all the external human pressures that we exert on the Baltic Sea. Right? And the question is if you want to use it to model. Well, that depends on your research question. If it has anything to do with body size, yes, you might want to consider this. If it doesn't have anything to do with body size, well, you can think about it, but maybe it's not the useful approach. Okay, thanks.